And as an example I gave, so I am the UK medicines regulator is called the MHRA. It's their regulatory body like the FDA in the US. And they have a series of committees of external experts. And one of them is a pharmacovigilance committee, which I'm a member of. So every time a risk management plan is written, people like me look through the risk management plan and decide, based on the safety specification, does this risk management plan look reasonable? Or if the company says we cannot do a study, it's impossible, I could decide, I think it is possible. So we would go back to the company and say, we disagree with you, we insist that you propose a study. And we could give them an example of what the study should be, but we can insist that they have to propose something different as part of the license. And this is something that happened quite recently in drug regulation, was that the authorities could refuse a license unless a commitment was made to do these kinds of studies. Okay? Right, so we're going to quickly recap on risk management, and then I'm going to go through several examples of where electronic health records are used for pharmaceutical research and why we would use them, how we make the best use of them, and what kinds of questions they can answer. And these are all real life examples where they've been used. Okay, so just to quickly recap. So the risk management plan has the safety specification, which tells us what we already know. So who's been studied, who hasn't been studied, how well does the treatment work, and what are the safety issues we already know. The summary of what we don't know, so the people that were not studied, any uncertainties we have about safety, so rare effects, long-term effects, anything we don't know about real-world effectiveness of the treatment, and then essentially how the drug will be used in the real world. What doses will be used? Will it be used off-label? So for unintended indications, that sort of thing. So that's the summary of what we already know and what we don't know. And then remember, the pharmacovigilance plan is where we say how we will address what we don't know. So how we will investigate safety outcomes further, and the risk minimization plan is how we will minimize those risks in future use. Okay? So I'm going to go through a series of examples now, which are opportunities where electronic health records can be really helpful for studying these various types of things. So we're going to look at investigating known risks, potential risks, filling gaps in our information. We can look at investigating effectiveness versus efficacy. We can also do randomized trials using the electronic health records. And these could be both pragmatic trials, but also trials where we use historic controls. And we can certainly use electronic health records to determine how well risk minimization is working. So these are all the various things that we can use EHRs to do. Now, one thing about terminology, I think it can be very confusing in the world that we work in today, where things get used a little bit interchangeably. So you'll hear people talk about real-world evidence, real-world data, routine care, non-randomized evidence, or big data. And lots of these will get used interchangeably. They're a little bit like a Venn diagram, where sometimes they involve each other, sometimes they don't involve each other. All I'm going to say is don't worry too much about these terms. They're broadly speaking, while I'm talking to you, I would say real-world data is data that's obtained during normal or routine clinical care. And that may well be stored electronically in large quantities that then becomes big data, okay? But don't worry too much about that kind of thing. That's the data. The evidence is what we might then generate using that data. So when we do a study and we produce a result and a conclusion, that's the evidence, okay? So, real world evidence. Hopefully, you realise from the first talk that because of this formal risk management process for medications, there's now a legally enforceable way of making sure that we generate evidence about what drugs are doing in the real world, okay? So, if we're going to do this kind of research about real-world effective treatments, where will we get the data from? Well, there are lots of different ways we could get the data. They're all related to how the drug is used in routine care, and you could get it from primary collection. So you could go to individual hospitals and get the records of patients and do studies. People do this. It's very resource intensive. It can be very expensive, and will usually mean you can only study a fairly small number of patients. We can have registries, and there are some great examples usually related to a specific disease. So for example, there's a rheumatoid arthritis register where all the patients taking the new treatments for that disease are all registered. But the two that I'm particularly interested in, because they're becoming used more and more, are your medical insurance databases and the electronic health records. It's also the kind of data that I use for my research. So I've put up here a few examples of data that could be used from either insurance claims or from routine care records, just to give you a flavour of the different kinds of records that can be used. So I've got some here in the UK. You've got the Clinical Practice Research Data Link. This is the database where I do most of my research, and we will talk a little bit more about that. The Health Improvement Network is another primary care data source, and Q Research. All of these are based on primary care in the UK. Denmark is an unusual case where the entire country is part of a linked record. Everybody in Denmark, when they're born, is given a number, an identification number, which then follows them throughout life. So their medical treatment will all go under that number. Their education goes under the number. Everything to do with their employment, their tax, everything is linked to that number. And in theory, researchers in Denmark only can access that data to do country-level studies where literally everybody in the country could be included. So it's a very unusual situation. Weirdly, the only records that you can't have to do this research on are primary care. So everything else, all their hospital records, all their prescribing, that's all available, just not the primary care data. But anyway, slightly weird situation. In the US, they rely very heavily on insurance claims data. So for example, Kaiser Permanente is an insurance database. And then here in Japan, you have a whole range of data sources which, who is using these data sources here in the room? Who's using any of these? Okay, a few of you are using these data sources. So if others that are not but want to know more about these data sources, you now know who to ask about them. I've never used them, but I think Masao may be able to tell you more as well. Okay. So we know, I showed you this in the last talk, that there's an explosion in the use of these kinds of data to do research. And again, this is the number of primary care publications using the CPRD in the UK. Now, a lot more of these kinds of studies will be done. And as I say, we need to be careful, but in lots of ways it's a good thing because it means that we can investigate these kinds of situations. And I've put these in, I've got bullet points, but basically the sorts of things we can investigate using electronic health records are drug utilisation. That's quite easy, and I'll show you an example in a minute. We can investigate safety signals, or we can investigate known adverse drug reactions. These three we've been doing for many years using electronic health records, and I would say that we've got very well established methodologies for doing it. We understand the limitations, we understand how best to understand the evidence and how to explain it to people. Where we are finding out how to do it is these four, the four at the bottom here, and I will mention these during the talk. So we've got signal detection, measuring drug effectiveness, randomized trials, and historic controls. And these are all growth areas in pharmacoepidemiology where the methods are not so well established, but where there is the potential for growth. Okay. So first of all, drug utilisation studies. Now I've got an example here of an assessment report published by the European Medicines Agency. So the agency will assess safety issues in great detail, and they will publish a document that describes all of the evidence that they looked at for this safety assessment. And they're all publicly available. It's a very interesting resource to have a look through. And I've given you one example here, which relates back to the question of sodium valproate and the problem with birth defects. So this is an issue that had been ongoing for several years, and there had been an agreement that what needed to happen was a pregnancy prevention program. Now, the drug is used in epilepsy, bipolar disorder, and migraine. And back in 2014, there was some action taken to try to restrict the use in women of childbearing potential and to prevent future pregnancy. So that's the risk minimisation. But the question is, how do we can have this program, but how do we know whether it worked or not? Because what really matters is that women did not become pregnant while taking sodium valproate. That's what matters, not that we had a plan in place. You see the difference? We could only investigate this using routine care data. So there's no point doing a randomised trial, because what we really want to know is, out there in the world, were women receiving valproate and becoming pregnant? That's the only thing that matters. So in order to do this, we need to have access to population level data that's suitable for doing this kind of research. And broadly speaking, for this question, we could split this up into three kinds of research. So three kinds of data. So first of all, we've got patient level data. We'd want to know the age and the sex of patients amongst our data, because we want to know about women of childbearing potential. So we're not interested in 4-year-old boys or 92-year-old women. We want to know about women of childbearing potential, so we need these demographic bits of information. We also need to know within our data, when did people enter the database and when do they leave the database? So in other words, a defined observation period over which we can find out what happened to these patients. Yeah. So that's your patient data. We also need to have clinical data recorded for them. So we would have diagnoses. I want to know, does this woman have bipolar? Does she have epilepsy? Why might she be taking treatments? So that's the kind of information I'm going to need. We also have signs and symptoms. Might be useful for us to know other things that are happening. And then really importantly, pregnancy. I want to know if a woman who was exposed to certain treatments has become pregnant. Okay. And then finally, I'm going to need to know what treatments people are prescribed. So I want to know the name of the drug, the dates that they were prescribed it, and potentially the dose that they were given. So if I've got within a data source all of these pieces of information well recorded, then potentially I can then use those data to do research, to investigate whether or not this problem with valproate was being addressed properly. Yeah? So what happened was, in the European Union, after they made these changes and these risk minimisation suggestions, they commissioned a series of drug utilisation studies in a range of different countries. So in France, they used a large national administrative database, and they were the leaders for this particular process. But they also involved other countries, including the UK, Sweden, Germany, and Spain. Now, there are 28 countries in the European Union, and this isn't all of them. And this is an interesting thing that happens in the EU, is that they won't do a study just in one country, but they will very rarely, if ever, do a study in 28 countries. So they will get an idea of what's happening in a range, but it won't be all of them. They largely used electronic health data in each of these countries to do this research. And what it confirmed was that after 2014, when these risk minimisation processes were introduced, the use of valproate fell amongst the relevant women, but the regulators didn't think it had fallen enough. They thought there was still more usage in women of childbearing potential than there should have been. And because of this, they then made further proposals for how to minimise the risk. So it was really important that they thought they'd made a good plan initially to reduce usage, but it turned out it wasn't the case. And only through doing drug utilisation studies could they then go on to suggest further improvements to those minimisation procedures. Okay? So that's one example of drug utilisation and why it's important. But there are lots of other reasons why you would do this kind of research. So why does it matter who uses the drug? Well, if you link it back to what we know in the risk management plan, we know about the identified risks, we know about potential risks, but there's lots of missing information. And this often relates to not knowing how the treatment will work in subpopulations. And we will only know if this is relevant once we get access to data telling us who is using the drug. So if it turns out lots of people with renal failure are taking the treatment, but they were never studied in the trials, then this is important information that's going to help us to decide what we might need to do next. It can help us to identify off-label use. So we know what indication we give the drug for, but is it what it's being used for? Are people giving it for other reasons? It can help us to identify drug interactions, so we can quickly see what treatments are people taking our drug in combination with, and might this be a problem? We can compare how similar or different the real-world population is compared with the randomised trial population. And we can then start to decide whether the risks and benefits might differ based on these characteristics. So again, similar to the example with the COX-2 inhibitors, where it turned out that in the real world, patients were much healthier than they were in the randomised trial. And then finally, as I've just shown you, you can also use these kinds of studies to see whether or not efforts to minimise risks have been successful. Okay, so I'm going to move on now to a study where we were investigating a safety signal. And this was some work led by a person called Anthony Matthews, who was at the London School of Hygiene. And this was a question about PDE5 inhibitors, these are drugs like Viagra, and whether or not they were associated with malignant melanoma. So there was laboratory evidence that suggested this may be the case, and then there were some conflicting epidemiological studies from both the US and Sweden. And that was our reason for doing the study, that we had this kind of uncertainty over the answer to this question. So we did a cohort study which involved comparing people on a PDE5 inhibitor with people not taking a PDE5 inhibitor, and then seeing who gets malignant melanoma in those two groups. Okay, so fairly basic study design. And I'm going to start by telling you a bit more about how you might use electronic health records to do this kind of research. So we were relying on primary care data. So how do we get primary care data and then me as a researcher try to make sense of it for a study? Well, fortunately, there are lots of databases available containing these electronic health records where we can do these kinds of studies. And the example I'm going to give you, because it's the one I know, is the Clinical Practice Research Data Link. So it's a UK-wide database which contains primary care data and a little bit of linked secondary care data. So we have some hospital data included in there as well. And it's been active since 1987. So we have potentially a very, very long period of time over which we can observe patients that were treated in the UK. At the moment, there are around 20 million patients included in the data set, and this is actually increasing quite a lot at the moment. So it's potentially a very large resource that we can use. So I've got the structure of the database here, which I've simplified a bit so that you can see the kind of information. So at the bottom here, we've got the patient. So on each patient, we've got the demographic information that you would hope to have. So the year that they were born, their sex, when they start and end their observation, their ethnicity, all that sort of information that you would want to be able to know about somebody before you do research on them. We have clinical files, which record information about diagnoses, signs and symptoms, and the dates that they happened. Therapy files record all the prescriptions that are issued in primary care. We don't have information on hospital prescribing. So if you're interested in drugs for, say, hypertension or statins, then this is great data because they are prescribed in the UK in primary care. If you want detailed information about cancer treatments, they're not going to be in there. So we can't do studies on the effects of cancer treatments. We've got test information. So if you've had various blood tests commissioned by the primary care physician, then those results will be available. If a patient is referred on to secondary care, then that's also recorded electronically. There's a whole range of additional information. So for example, whether or not people smoke, how much alcohol they drink, their body mass index, all of that sort of information is recorded. Obviously, it depends on how honest the patient is. So for the BMI, that's fine because the doctor will measure it. Smoking and alcohol, well, people will say what they think the GP would like to know, or the doctor. So it may not always be honest, but there'll be some degree of accuracy there. Okay, so that's the sort of information that's recorded in the CPRD. This will be similar for other databases, and there will be some differences as well. Okay, so we've already talked about this in the patient file. Don't think there's anything else in there that you need. We also know if people die. So we have the date of death, which is very useful to know. And also we have their socioeconomic status. So these are all variables that if we are worried about differences in patient groups, that we can potentially adjust for. Again, that's clinical files, which I've mentioned. I don't think there's anything else. Nope. Therapy files, no, again, we've talked about that. I guess just to point out the sort of information can be quite detailed. So as well as knowing what drug the patient was given, you can have things like the quantity of tablets they were given, the number of daily doses that was expected to translate into. But this can be quite variable in how it's recorded. Sometimes it's missing, sometimes it's there, and that can be problematic. So that's the data, which sounds quite involved and very detailed, but how do you get started to do a study using these data? So if we want to know, do drugs like Viagra cause malignant melanoma, where do we start? Well, it typically involves identifying people based on either an exposure, so a PDE5 inhibitor, or an outcome, like cancer, or both. Now, to do this, we need to develop things like code lists. So there are many different PDE5 inhibitors available, different strengths, different products. So we need to know the code for every one of those possibilities. Okay, so we need to put that list together. Drugs are relatively simple to make code lists for. Diseases can be very time consuming. So if you go to the GP and you've got cancer, the GP will enter a code onto their record for you to say what kind of cancer you've got. Now the GP is free to choose from a massive range of codes, and they will use whatever is most convenient for them to keep a record of you clinically. That's the purpose of the record, is for them to manage you clinically. Unfortunately, the purpose of the record is not for me to do research. It's a happy accident that I can use those data to do research, but we have to understand it's not the primary purpose of the data. So it means that if I want to identify cancer using the CPRD, I'll need to develop a really detailed code list that potentially captures every kind of cancer that could be recorded. And so, for example, if you take breast cancer, there will be many ways that the GP could record that a woman has breast cancer. So cancer has over 3,000 codes that could be selected in the CPRD. And we as researchers have to put together that list. So that's just a list for cancer with 3,000 codes. If you want to take any other disease, they will all also have similarly very detailed code lists. So it can be a very time-consuming process to put these lists together. If you get 2 investigators, they won't all agree on what should be in each list. And there will be some really obvious ones. So there'll be a code that just says breast cancer. Now everybody will agree that code should be in. But then there will be some obscure, unusual terms that are slightly ambiguous, and we need to decide, do we include them or not include them? And there will be disagreement. So I say that so that you can understand when somebody presents you their study result and says, here's the effect of this drug on cancer, that there is some kind of what I like to call fuzzy edges over how we define these illnesses. And you need to be aware that this is what we as investigators are doing all the time. Okay. As well as doing this for our exposure and outcome, we also have to do it for other patient characteristics. So other comorbidities, other treatments that they take, but then things like the smoking and alcohol information. Now, smoking is something that the GP, the primary care physician, will ask a patient about when they register. And the GP, the patient may or may not lie. They may tell the truth, they may not tell the truth, but the doctor may continue asking them over the years. So if you look at a patient's record, on the first day, it may say they're a smoker. 2 years later, it may say they are an ex-smoker. 5 years later, it may say that they're a never smoker. Now, all of these things cannot be true. So we as investigators have to decide, how will we interpret that record? Personally, I would interpret that they were a smoker at the start and then became an ex-smoker, and that I would never believe that they were a never smoker. But that's a fairly simple example, and it could be much more complicated than that. And you have to decide at a given point in time, what's my best guess at whether this patient is a smoker or not. So again, an example of why the data are complex and can be difficult to use. Okay. So back to our research question. So we wanted to know whether or not PDE5 inhibitors cause malignant melanoma. So we had 145,000 men taking a PDE5 inhibitor and 560,000 without. Now, we need to think about, so we want to know what's the causal association between these two. But the trouble is, we have to think about other things that may confound the association or may also explain any association between PDE5 inhibitors and melanoma. So as researchers, we thought men that take PDE5 inhibitors will be different from men that don't take them. It could be that they smoke more, it could be that they drink more alcohol, or it could be that they have a higher body mass index. So lucky for us, we've at least got some information on these factors that we could include to adjust for. But any thoughts on what else might differ between men that take PDE5 inhibitors and men that don't, and which is highly relevant for whether they get skin cancer? What might explain that association? What causes skin cancer?
Sunlight. Exposure
Yeah,
to sunlight.
sunlight. So, is it possible that men that take PDE5 inhibitors are more exposed to sunlight than men that don't? It's possible. We can't measure it. It's not in the GP database, so we don't know about that. So we can't adjust for it. Now, I don't know whether on your slides, whether you can see this better. These are the results from the study. And so what we found was that men taking PDE5 inhibitors, even after we adjusted for everything we had a 14% increased risk of having malignant melanoma compared with men who didn't take PDE5 inhibitors. Okay. Now, we weren't certain that this was causal, and so because we were worried that sunlight exposure may differ between the groups, we also looked at some other outcomes. So we looked at basal cell carcinoma, which would be another marker of potentially more sunlight exposure, but would not be a marker of something that would be causally related to a PDE5 inhibitor. So the mechanism would not have affected both types of skin cancer. And so here you've got also an increased risk of basal cell carcinoma amongst men taking PDE5 inhibitors, a 15% increased risk. So we thought this was some indirect evidence that the association with malignant melanoma may not be causal. And then finally, we also looked at the association with solar keratosis. But we looked at this association before the men had ever taken a PDE5 inhibitor. So before they'd ever even had that exposure, they also had an increased risk of having previously developed solar keratosis, another skin disease that's linked to sunlight exposure. So we took this as being evidence that we ought not to interpret this increased risk as being causal, but that in fact there seemed to be good evidence that men that take PDE5 inhibitors actually are more exposed to sunlight, and that this is probably the explanation for this association. Now originally this could have been presented as, look, people taking Viagra are getting malignant melanoma. Viagra causes cancer. But of course we want to be really careful that this isn't the sort of message that we're giving when in fact, when we look more carefully within our data, it's telling us what this association may be due to. Okay, so that was an example of electronic health records being very useful to investigate a safety signal. Now, so investigating safety signals and drug utilisation are things that we've been using these data for years. So I want to then move on to what I see as the future in using real-world data and evidence for pharmacoepi research. So starting with safety signal detection. Now you know from the last talk that one area where we use spontaneous reports is to develop hypotheses of drug safety signals. And we're very used to doing this using disproportionality. But the question is whether or not we might also use the electronic health records themselves to generate signals. And I'm not going to go into detail on the methodology, but just to give you a flavour, there's a partnership from a few years ago called OMOP, the Observational Medical Outcomes Partnership, and this was a large public-private partnership that was led out of the US, and it looked at healthcare databases and how we could use them better to study the effects of medicines. There's a great website which I've given you details of there, where you can find more information about what they did. But what they did was looked at whether or not we could use electronic health records to look for signals of adverse reactions to do with vaccines. And then more recently, we've also done some work looking at whether we might use them to investigate the effects of non-vaccine medications. And much of the methodology is around the use of self-controlled methods, which I'm not going to go into detail on now, but I will tell you more about on Friday. Okay. And moving on to, there was a paper published back in January by Jesse Franklin from Harvard. And I think this is a really nice summary of where we are at the moment in terms of real-world evidence and where it can be used and where we are still trying to develop the methodologies. So there's the safety bit at the bottom here, which I've just been talking to you about, where we know that we can use real-world evidence to look at the effects of medications. But what's really interesting is this bit here, where we wonder how and if we can use real-world evidence to investigate the intended effectiveness of treatments. So you've got here, we're going to talk in a minute about historical controls. You've also got the idea of secondary indications. So whether or not, once the drug is approved and it's then on the marketplace, can we ever use real-world data to approve indication extensions? So for example, could we extend an indication to children even though they weren't studied in the randomized trials, purely based on real-world data? And I would say at the minute the answer to this question is not known, but there's the potential that we could. And then there are also what we call adaptive pathways. And examples of this might be where there's a high unmet medical need, and so a treatment is given a license quite early on, perhaps because it's shown an effect on a biomarker, when what we really care about is a clinical endpoint. And so whether or not, once the drug has been approved, we can then use real-world evidence to supplement what we know from the trials to look further at clinical endpoints. So I think all of these are quite interesting, and I would suggest that if you're interested in this, read this paper. It's a really well-written paper that talks about the future of where we might go. But I'm also going to talk a little bit about where we are, or where I think we are, in electronic health records for effectiveness. So it's built into the legislation in the European Union, at least, and in some other regions, that knowing about treatment effectiveness is very important, and that we can insist that companies do studies to look at effectiveness in routine usage. Whether or not we can rely on observational data to do this is an unanswered question, and whether or not there's too much confounding in observational data is uncertain. And I would say, if it's possible for us ever to do this, it might be in quite limited circumstances. I don't think there's ever going to be a wholesale move to investigating effectiveness in routine care data. But we're still trying to decide if and when we might do it at all. Having said that, there is the possibility of actually randomising within routine care data. So the idea of pragmatic trials was raised quite early on, where we might want to know, within just normal care usage, if I randomise people to one treatment versus another, what will happen? What will their outcomes be? And that this might be quite different to what we see in the pre-marketing explanatory trials, where everything's perfect, where care is perfect. We pick our patients because they've got no other diseases, no other medications, and we see our perfect efficacy. But there's no reason why we can't just randomise people in routine care to also receive a treatment. And I'm going to move on slightly. One of the things that we can take advantage of is the fact that doctors themselves can act a little bit randomly. Now, it isn't always clear to a doctor when you might give one treatment versus another. Maybe there's no good reason why, if you've got a patient with type 2 diabetes and they've already tried one treatment, why would you give them the new treatment versus a sulfonylurea or an SGLT2 inhibitor? How would you choose between them? Well, sometimes they don't know. So in fact, it's possible that you could take advantage of that indecision and just randomise people to one or another treatment. So it's harnessing uncertainty that already exists clinically and then making better use of that to be able to then determine the real effects of treatments. Here's a trial that was done a few years ago within the Clinical Practice Research Data Link. And the idea was that it was a pilot for future trials, just to show that this was a possible thing to do within the database. And it was basically looking at randomising people to either atorvastatin or simvastatin. So the patient definitely needed a statin, and then the GP could randomise them to one of these two statins, which you would think was a fairly simple thing to do, a fairly simple thing to get agreement on, and that the regulatory hurdles would be low in order to do this kind of study. The way it worked was, if the patient was in a GP practice and the GP was about to prescribe either atorvastatin or simvastatin for the first time ever to that patient, there would be a screen that popped up on their prescribing terminal to say, this person could be in our randomised trial, do you want to recruit them? The GP gets to decide, I'm a bit busy, no, go away, or yes, this looks possible, I'll talk to the patient, we might recruit the patient into this trial. And then the beauty is that the system would then randomise the patient to one of the two statins, the doctor would make the prescription, and then nobody would ever need to do anything else for the trial in the future, because all we would do is simply collect the information from the electronic health record as it happens. So it wouldn't rely on the doctor filling out forms or remembering to do something. In fact, we wouldn't want them to do anything different for that patient. Just prescribe the treatment and just follow them up as you would normally. Okay, that's the beauty of this. Would then generate real evidence of what happens in routine care when a patient takes one treatment versus another. Yeah? So it sounds nice and simple. Sounds like it should have been easy. But in fact, what happened was there was a massive dropout rate. So there were 459 individual primary care practices that agreed or that were contacted to see if they would like to be in this study. Over 80% returned the letter to at least say whether or not they were interested. So 18% didn't even reply. 58% said that they were interested in being a part of this kind of study. So that's quite good. The problem is, as they went down all of these regulatory hurdles that they had to go through simply to prescribe either atorvastatin or simvastatin in the context of it being randomised, meant that they mostly dropped out. And we thought this was fascinating, that you take a very non-controversial question like atorvastatin or simvastatin, and suddenly you find yourself in a position where everybody in the practice has to be given good clinical practice training for being in a trial. Well, the GP's just deciding for themselves normally whether to prescribe atorvastatin or simvastatin. Nobody insists on coming to give them any training when they make the decision. But if they roll a dice, then you have to give them all this training. So what the investigators found quite depressingly was that in the end, only 17 practices out of originally 459 were able to recruit any patients. So the main reasons that they gave as reasons why they stopped taking part were the time and workload for the clinician. It was just too much, and the ethical and regulatory approval needed seemed unnecessary for this kind of research. So there's a lot to do in terms of having this kind of process embedded within a health system, but I am still quite hopeful that there will be progress. But just to say, it's not there yet. Okay, and then moving on to a study that I'm involved with, again in the CPRD, where we're looking at the real-world effects of medications for chronic obstructive pulmonary disease. And this is the published protocol. And we're trying to use the electronic health records to answer a question about effectiveness, but using the results of a randomised trial to validate our findings. Okay, and the way that this works is that we start off with a real randomised trial that was done some years ago, and we already know the results. We can access the individual patient data for that trial. There's a website that you can apply to called clinicalstudydatarequest.com, and you simply ask them, you tell them what your research question is, and they grant you access to the data. So having got those individual patients, what we then do is go to the CPRD and find patients who look as similar as possible to each of those patients in the trial. We then look within those patients who are very similar to the trial population to see if we measure the effectiveness of COPD treatments amongst our CPRD electronic health record population, do we get a result which is similar to what they found in the randomised trial? And if the answer to that is yes, we see a similar result, then our next step would be to ask questions about the sorts of patients who were not included in the trial, maybe because they were too old, or they had more severe COPD, or whatever the reason was, and then try to estimate the effectiveness of COPD treatments in those patients, given that those are clinically interesting questions where we don't know the answer. Okay, so we're still working through this study. We are currently at this point And all I can say is things are looking good so far. So we'll see how we get on with this section of the study next. But it's another potential way of using the electronic health records to try to estimate treatment effectiveness. My question, which I still remain with, is will anybody ever believe the answers that we get from a non-randomised comparison of these records of treatments? And I don't know the answer to that. You might have your thoughts. I'm sceptical as to how often we will believe the results, but I'm open to possibilities. But it is, like I say, it is very much where we are in terms of methodological development right now. Okay. I think there are some ways that we can think of approaching it. So we can compare our results with trials, where trials are already available. We can have control outcomes or exposures, a little bit like we had with the malignant melanoma study. So within the data, asking the question, what do we know to be true already, and which we could demonstrate with the data? And then the last thing we can do is something called quantitative bias analysis, where we take our results that we generate within the observational data and we ask a question of, if there was a confounder that we were missing, or a series of confounders that we miss, how strong would they need to be to explain away the result that we have? And if the answer is, well, if you'd have just had slightly better control for confounding, you would have had a null result, then I won't believe my results very well. If the answer is, you would need to have an enormous amount of confounding to change the results that you generated, then I may have more confidence that my results are correct. But these are things that I think we need to do, but I think we still don't have a good idea how best to do them and how best to interpret them. Okay, and then I mentioned earlier on the idea of using historic controls. And again, this is a recent idea that we might be able to use electronic health records to provide controls for current randomized trials. So if you imagine that you've got let's say that there's a population of people with cancer, and we've got a new treatment, but which we think possibly doesn't work well for everybody with the cancer, but might work on a small subgroup of people who are red. If I do a standard randomized trial in everybody with the cancer, then the clinical effect size that I see will be small, because the drug's only going to be effective in a small subpopulation. Yeah? So I could do my big randomized trial, but it isn't going to be that useful. What will be much better is if I can do a randomized trial where I only include the subgroup who I think might benefit. But the problem is, they might be a very small group of patients, and it might take me a very long time to recruit enough patients to do a randomized comparison against either the next best treatment or no treatment, whatever it happens to be. It's possible, if the data exist, that I could use patients that look like these red patients, but maybe 3 or 4 years ago, when this treatment wasn't available, if I've got the right kind of data. There are lots of considerations that we need to be careful about if we're going to do this. So we need to be careful exactly who we would compare with. What sorts of patients were they? Are they easily identified from a data source that already exists? And it would be really important that we capture all of the relevant information for those patients, such as what disease stage they were at, what their outcomes were. So this is not straightforward, but it's the sort of work that people are doing to see whether we can use historic controls to use as the non-treated comparator arm for current day randomised trials, if that makes sense, or controlled trials. So it would be quicker because more eligible patients are treated, which then means that potentially new useful treatments can be given to a wider group of people quicker. But there is a risk that we will get the wrong answer because we're relying on this previously recorded information, which may not be perfect. Okay, so to summarise, so we know that electronic health records are being used more and more. We've got some very large data sets from all sorts of regions of the world. I should also mention that there are, of course, big regions of the world where these data are not available. And if you're working certainly in lots of countries in Africa, for example, it's very rare to be able to access data on this kind of scale. And in fact, the treatment effects you might see in some of those countries could be very different for good reasons. And we won't be able to answer questions on them until we've got access to better data in more regions. Our understanding of how to make the best use of these data is at different stages depending on the sorts of research questions we want to ask. So we have well-established methodologies for looking at drug utilisation and to estimate the risks associated with taking drugs. Where we are still finding out the best methodologies and whether or not they're appropriate are areas like signal detection, looking at the effectiveness of treatments, doing randomised trials within databases, and also using historic controls. Okay, so I've got a range of references there that you might find useful.